It's an honor to be here and to speak, and it's very hard to speak after this testimony. Again, I share your frustration. For me, when I started as a physician, I want to help. I really want to understand. I want to really put any effort and everything that I can into making things better. What the first presentation that you said, be humble, get expertise if you don't have it. Sometimes there is no expertise. Sometimes you need to build that expertise for the others and for the others to go forward. But again, the thing is, as a physician or even as any human being, the best way that we could do to help people is by initially listening and trying to understand. For me, research is about understanding. Sometimes it brings me closure. It brings family closure, even if for some reasons, for now, it doesn't help their kid. They know that whatever we find will have other children, other human beings. So for me, being a physician is great, but being a researcher is equally important. Zero translates into zero. If I want to help, especially in rare diseases, especially in the cancer that I treat, I need to understand better. Up to now, in the cancer that I treat, the brain tumor that I treat, zero child survived more than 2 years. So I have to do better. We have to do better as a community, and this is where I started. I'm a pediatrician, so I'm a storyteller. So I'll tell you a story about how a snail became a big jaguar. When I started medicine, I heard through someone about the PCR. And for me, it was fascinating to see that an enzyme could function at such high temperature. And this is actually how we were able to clone our DNA, to sequence it. And I wished, and I really dreamt that one day I would be at a time where a revolution such as the one that brought PCR and Sanger and brought us our DNA and our DNA sequence would be here. And I was lucky, and I got very lucky, because I came at the right time where all of those tools that are giving this big data that you're speaking about came about. And this came through the revolution of the next generation sequencing. Just to give you an idea of what it is, to sequence one human genome and the sequence what put forward in 2000. It took about, say, 20 years. I don't know how many, 250 Sanger machines, one after the other. I don't know how much microgram of DNA for one genome and a cost of $3.5 billion. Do you know how much I can sequence in 2 days today and for what cost? 96 genomes, 2 days and a half, $800, and the cost is going to plummet. And initially, the resolution was barely, if you remember, when you were downloading and you were waiting for this thing to go down. Well, now, you have the resolution to no end. And that's important, because an organ, a tissue, a tumor, is made not by one cell, but multiple cells. And even we share, all of our DNA is probably the same. There are sequences that change and shift. And actually, those changes are the ones that create disease. And this is the best way to sample this heterogeneity, this diversity within a given sample by sequencing deep. And this is what happened. And this is how now we are actually seeing, and especially rare diseases, it helped a lot. Initially, it would take, I don't know how many families, the lot scores, whatever, to try and define the area of the genome where potentially the gene would be. Now, even with one patient, you can identify stuff. And you could make sense of things that didn't make sense before. I want to put a word of caution. DNA, we all, when I have 3 kids, when I see them, I say to myself, my God, what is it? What's going to happen to them? I don't want anything to happen to them. But so we want to know what is it that we were born with, what our children are born with. And people say everything is in our DNA. And I hope that this DNA never, ever, ever segregates us. For those that are old enough like me and have seen this movie, GATAC, TCA, like our DNA sequence, where people are actually completely discriminated by their genetic information. This is a society that I would never want to live in. Understanding and providing better care through understanding of our genomic sequence is great. And it should be for that purpose, not for discriminating any of us. And please keep that in mind. So, pediatric brain tumor, my focus, my story. When I came, I'm not a good example for medical studies. I started as a surgeon. I love surgery. And then somebody told me I would be bored. I went into research, did immunology, hematology, pediatrics. And pediatrics were almost by mistake And then I married a Quebecois and came to Quebec. And in Quebec, there are not a lot of immune deficiencies. So I looked, and brain tumor seems like very orphan, especially in kids. Nothing was known. Those children die. And I'm an oncologist by formation, so I adopted that disease and started using the tools that I was taught to into practice for them. And we hit gold. And this is another thing. Never work alone. It's a collaboration. And this story that I'm going to tell you is a story of multiple people across multiple countries, within my own institution, outside of it, where we put our efforts together, our samples together, our families together, and made sense. And this is a paper that was published in 2012 and had a lot of highlight. This is one of the most highly cited papers today. Why? Because it actually showed that one gene that never ever anybody could think would be mutated got mutated, and it's a histone gene. And I'll tell you the story of that histone gene. This finding was discovered by Jeremy Schwarzentruber, who was a master's student, co-supervised with Jacek Majewski, a dear friend and colleague of mine. This is his discovery as much as it is mine, in collaboration with Stefan Pfister from Germany, and Xiao Yang was an MD PhD student at that time, and she helped with that. What we did, we identified 2 somatic mutations. Somatic meaning they're not in your blood, but in the tumor, that change a lysine at position 27 to methionine, and a glycine at position 34. You don't care about this, just 2 mutations. And know that the lysine 27 in a histone gene, it's your mom at house. Whatever she says, you do. And actually, this is what dictates a lot of the things that the cell needs to do, a DNA needs to do, and you'll see why. This gene that we identified to be mutated is so conserved at the amino acid level that even plants, this is Arabidopsis thaliana, have it, you know. This tells you that this is a gene that you need not mess up with. And those 2 residues are so conserved. It's impressive. So what does a histone do? Our DNA, 2 meter 10 long, completely compacted into nothingness into the nucleus, 0.2 micron. The boring but very important task of histones, who form the nucleosomes, 147 base pair unroll, and then you trill, compact into nothingness. And then this DNA needs to make sense, because this is what makes us. This is, the genes are there. They need to be accessed and whatever. And this is the beauty of the nucleosome. It has this histone have tail that open and close, make our genetic information accessible when it needs to be accessed. And unfortunately, this is the problem that happened for those kids. With this histone tails, when we got the sequence of the genome, the first genome, 22,000 genes, exactly like the mouse. And we think that we are a little bit more advanced than the mouse or the chimp. There is barely any diversity with the gorilla, with our next of kin. It's maybe not the amount of base pair of genes, but the way we play with our genetic information that makes us what we are and who we are. And actually, we've evolved to have tools, to have with the same DNA sequence, so many different possibilities, so many different genes, and within a gene, so many different isoforms to create so many different proteins that make our phenotype. And the way to do that, to dictate the information to this genetic information, is the epigenome and those histones. And we've evolved for those histone tails to have something that writes a mark, something that reads a mark and act on it, and something to erase it, because you should not, it's one task. Once it's done, you should not repeat it. And this is how we've evolved, to play even better with our genetic information. And maybe the best way to say it is think about our DNA and our genetic information as notes of music. Do, re, mi, fa, sol, la, si. Do you know how many different types of music have been made just with this? Well, this is what the epigenome does. It partitions our genetic information. It gives sense to our cells. Actually, we have the same genetic information in every given cell that we have. But you agree with me that a fetus is different from an adult. A brain is different from a heart. A heart is different from another muscle. And it's a way we open and close our genetic information that makes us what we are. And this is the partition of life, and the epigenome is putting it. And unfortunately for those kids, the partition is very wrong. It is the barcode of life that's stuck, and you're providing non-harmonic graph. This is the way I say it. and you're stuck in a way that you're not going. And not everything is in our DNA because the epigenome is very important. When I started this, how it looks, nobody was interested in histone or that much. And this is exactly what it looks now. A lot of people, and that's good, because when more people are working on something, a cure will happen. Things, knowledge will advance and will go and will be better than what we are. And what we did is we showed exquisitely that those mutations are very tightly associated with brain development. Based on the age of the patient, the location of the tumor, I can guess exactly what they are. 3 to 5 years, this is, so combination is going to be 5 to 7 years, still the pons, this is what it's going to be. The thalamus will have this combination at that age time. The spine, this is where it's going to be. The temporoparietal cortex, this is what is, the other mutation is going to be there. And the frontal cortex, this is what's going to be at that age. Just by looking at the MRI and knowing the age of the patient, I can guess 9.9 times out of 10 what they have. This is brain development gone wrong. And every tumor has its story, and I can find it. I could identify what it is. And those are 2 lysines, one lysine 36 and the lysine 27 that I told you about, that are wrong and acting wrong. And this is a new paradigm of cancer. So what we said, this schema is just to show you that this is a yin-yang between a very repressive mark, the lysine 27, when it's methylated, and you see why I'm bringing that up, and the lysine 36, when it's methylated, that's activating. One is saying open, the other one is saying shut, and it's not working to those patients. And what it's creating actually is what I call the Peter Pan syndrome. What we've shown exquisitely is that those mutations actually make your progenitor cell, it's every cell that we have from the embryonic stem cells is going to differentiate into brain, into a heart, and then within a tissue, it's going to differentiate into the different cell types that it needs in the brain. You'll need an astrocyte, a neuron, other type of an oligodendrocyte to create through your myelin. And each step that you go, you can't go back. If you do it too early, this differentiation, you don't have enough cells to create your organ. So this lysine 27, when it's methylated, it promotes differentiation. So what happens if it is promoted too soon, you don't develop well. If it's not prodded, you stay in the same cycling type. You don't go forward. And this is why I call it the Peter Pan. This mutation actually makes this lysine 27. Doesn't get trimethylated as it should be. So you never, ever, ever go and further differentiate. So you have a mini brain organ that thinks it's going to grow and grow and grow, and those are the tumor that we see. The other thing is the data, when you were speaking about who owns the data. There was a beautiful paper that is redacted, as you see in the New England Journal of Medicine a few years ago, where the authors were saying that we researchers are parasites because this is data that should be owned by the researcher and not shared. Well, I beg to differ. The data belongs to patients, to taxpayers, and to people that really can make sense. When you do it, put it out in the open if you have permission from the patients to do it this way. Because no one person can make sense of it. But a lot of persons can make even better sense, and I'm very proud to be a research parasite. And what we did is we made sense. We found other mutations. This is not my work. This is another group that found other mutation in 2 tumors, chondroblastoma and giant cell tumor of the bone. Those histone mutations were identified there. We identified it in acute myeloid leukemia. In 2%, they have the lysine 27M mutation. We identified in 10% of undifferentiated sarcoma the lysine 36M. And for pediatrician, in 15% of head and neck cancer. Also mutation in this. So you have 2 marks, one suppressive and one that's activating, that are opposing each other, that are driving those cancers. We need to understand them better. We need to really work on that. So we're doing mouse models. They're not easy, but we're doing them. And any model actually is good because then you could potentially tease out what is it that you could do to treat. We're even going with the flies, regardless of what people can say. And I don't know for those who remember Sarah Palin, who was appalled by the fact that somebody would be working on flies. Well, flies, when you put those tumors, those mutations, they have small eyes. After that, you could run modifier screen on the eye of the fly, and you see what is it that rescue it. You're not touching patient. You're not giving them anything that's toxic. but you're making them benefit from whatever that you can find out there. Because this is what we want. We want the targeted therapy to kill the bad cell, not the right one. And the challenge is that all of those have costs to that. Who pays for those sequences? Who pays for this? Who pays for the care, for the cost of that medication that's going to be in an orphan disease? In the case of the oncohistone, the fact that you're obviously having it in head and neck cancer and other sarcoma makes it a little bit more attractive for pharma, who really wants not to be treating small little 20, 30 patients here and there. But that should never be an excuse. Even if there's one patient and there's a possible cure, there should be efforts and money and will to go and do forward. And this is why we need to work together as a community to that. And I hope I've convinced you that we're coming from here, and I'm a Trekkie. I hope that we're going to be even faster than that. And then the WHO classification for brain tumor came, and everything that we identified, they adopted the K27M. But there are so many different other markers that allows us to better stratify and to treat more exquisitely patients. It's going to take some time. Remember what I told you about having people embark on it? Well, we're lucky. Even Hollywood embarked on it. My kids never thought I was funny or anything until we were looking at this amazing Spider-Man. I don't know if you noticed, this is a screen capture. And whenever I look at those black boards, I was wondering what Tony Parker was saying to Dr. Connor, is a future lizard, if you remember in that story. What was there? Well, even Hollywood knows about the lysine 27 trimethylation. So if I didn't convince you, well, they were convinced. This work was done by the people in my lab that I love dearly, those students that are going to become, the researcher of today and tomorrow. Jacek, who is really an amazing collaborator, everything of my story has been with him. Claudia, who was initially a postdoc in Jacek's lab and is being an amazing worker here. The different people across the world. This is never a sole initiative. You start with an idea, but then you embark and you have other people embark with you if it's worthwhile. The funding, and it came. And I do this because I care. Thank you very much.